All right, everybody, I've begun the recording. It is September 26th, 2025, and this is Biology 105 lecture. Thank you all for being a part of it, because you can have a Bio 105 without me, but I can't have a Bio 105 without you. Anyways, it makes sense, I think. Here's what we're going to do, all right? We're going to wrap up cell structure and function today. That'll officially finish our week 4 material, and my fingers are crossed that we will be able to begin our week 5 material. Don't worry, the offset of lecture weeks is regular thing that I've adjusted for and planned into the syllabus, so no need to panic about that stuff. Continue doing your readings as scheduled, continue doing your reading quizzes, all that good stuff. You did labs this week, it's Friday, so we don't have to worry about that. We're just gonna do mainly lecture today. I don't think we really need to do any other stuff about the course structure. Cool. Enable content. Great. All right, I wanna make an official announcement just saying thank you all for your continued involvement in asking great questions so far this semester. It's not always that I get such good engagement, and so more questions, more questions, more questions. If you ask the question, rest assured there are 5 to 10 other people in the room having the exact same question. So I just want to say thank you all for that degree of participation in the course. It really makes it more dynamic, and I'm just really grateful. With enough of that out, let's get that out of the way. Okay, no more positivity. I'm just going to get real grumpy and negative as we talk about endosymbiosis. I believe this is where we got to on our last presentation. Let's go ahead and continue there. All right, so this is where we stopped. If you'll remember, there was a free living organism. That free living organism developed a nucleus, okay, by infolding its plasma membrane. Now, this protected its genetic material. It tried to eat a bacteria that could carry out photosynthesis. It also tried to eat an organism that was very good at carrying out cellular respiration. Instead of eating these things, maybe it had some mutation in producing the enzymes inside of its lysosomes. The things continued to live and lived on inside of the cell. As the cell divided and made more offspring, they got some of those little endosymbiotes carried along with them. This is the process of endosymbiosis. There's a lot of evidence for it, including things like some genes from these organisms have been transposed into the nuclear genome, right? These things can no longer live freely. Their membrane composition and structure is slightly different, all right? So all evidence points towards this being the evolutionary origin of the organelles, specifically the chloroplasts and the mitochondria. All right? Cool? Questions about that? Endosymbiosis? Symbiote within. And there are plenty of other examples in plants and animals, especially in plants, of endosymbiotes and endoparasites, things that live inside of cells and provide a benefit or are parasitic. Vacuoles. I've already talked to you about these. Vacuoles are big vesicles. What's a vesicle? A vesicle is a plasma membrane, it's a phospholipid bilayer. It's a little sac that contains some stuff. A little vesicle's a vesicle, a big vesicle's a vacuole. All right? Vacuoles are big, so they tend to have more active roles in the cell. Like the central vacuole is incredibly important, and the contractile vacuole is incredibly important. Yeah. So food vacuoles, contractile vacuoles, central vacuoles. Phospholipid bilayer holding something. We know what a food vacuole is. We talked about it. We talked about phagocytosis. Okay? Contractile vacuole. We talked about it way back in week 1, I think, when we introduced you to the protists. But now my goal is that this should make a lot more sense because you have been talking about osmosis in your lab, right? So contractile vacuoles are found in many freshwater protists. A protist living inside of fresh water, well, it's in a hypotonic solution. There's a higher solute concentration inside the organism than outside the organism. Therefore, water flows into the organism. Too much pressure inside, the cell could burst. So a contractile vacuole is basically, you know those little ear cleaning bulbs? You know what I'm talking about? Little blue bulb with a thing. It's like one of those attached to the outside of the cell, where it just absorbs water from inside and goes and just squirts water out of the cell every once in a while. All right? By contracting. It's a contractile vacuole. I keep it on the slide because I like to talk about osmosis, and this is another time to think about why would it only be in fresh water, not salt water, organisms. Anyways, central vacuoles, we've talked about them. Central vacuoles are important inside the plant cell for maintaining turgor pressure. They also hold organic compounds. Okay, it's a big, broad statement. It's a big storage compartment, and it can be used for developing turgor pressure. There's a ton of other stuff, of course, that it's important for. But this isn't even Botany 105. This is Bio 105, so we're not hardly going to talk about that at all. Endomembrane system. We talked about it already, but I am using repetition to emphasize important concepts. All right, you've been in school before. You know we do this. So if you have any questions about this stuff, just slow me down, let me know. Otherwise, I'm going to assume that the repetition is serving its purpose to, make your brain go, oh yeah, I remember that. And every time your brain does that, it gets more cemented in your brain, right? So endomembrane system, right? It's all connected, main thing, it's all phospholipid bilayers. They can fuse, break apart, Cool? Oh, all right, here we go. We're continuing to move forward. All right, mitochondria and chloroplasts. They're membrane-bound organelles. They're complicated membrane-bound organelles that are a little bit different than the members of the endomembrane system because of their origins as free-living organisms. Okay? They used to be free-living things. Now it's an obligate symbiotic relationship where they cannot live without the host cell, that being the animal or plant cell. What they can do, though, is divide and reproduce. However, some of the genes essential for their function are stuck in the nuclear genome, so they don't even have all the genetics that they need to survive forever. So the mitochondria, the chloroplasts, can't live freely anymore. They're entirely reliant on the host cell, okay? So they depend on other parts of the cell for some of their internal components. We are not going to talk about peroxisomes. Don't bring it up. I don't want to think about peroxisomes. Thank you. They're the worst, okay? Mitochondria make a bunch of excess energy. Good stuff, right? Good energy processing. Carry out cellular respiration super efficiently. Chloroplasts carry out photosynthesis, provide the cell with high energy organic molecules. Energy just goes from solar into here, makes chemical energy. Chemical energy gets broken down, does the cell stuff. Cool? I'm stoked. I'm having a great time today. We are destroying this. It is Friday. Life is good. Life is good. Okay. Mitochondria, chloroplasts. Here's the main common thing you're going to see between them. They've got this weird, super folded up membrane. Both of them have a weird, super folded up membrane. Why? To increase surface area, because there are a bunch of important things, proteins, embedded in that membrane, all right? And the main energy processing is going to occur across that folded up membrane. Cool? All right. What do mitochondria do? Oh, they're found in nearly all eukaryotic cells. Great. They have an outer membrane, which is smooth. Let's see, do I have a good marker by any chance today? Yeah, all right. They have an outer membrane, which is smooth, and an inner membrane, which is folded. All right? So it looks like this. You don't need to know, but the folds are called the cristae. All right? But here's the part that you do need to know. Whenever something gets divided, right? Think about a house. A house that has no rooms in it. You go into the space and it's like, well, this is the house. But once you start to divide it into special rooms that have a special function, now you say, that is the bedroom. That is the bathroom. That is the mud room. That is the pud room. That is the chud room. You know, that's how the rooms divide in a house. Everybody has those, right? Okay, so you divide something. Here's my point. Once you divide something up, you give the different chambers names. You give the different rooms names, and then we can actually talk about it. You know what I mean? So I can tell you, like, the bicycle's in the house, or I can tell you the bicycle is in the closet in the bedroom. That's way better, more accurate. So the words we use for orienting ourselves into with the mitochondrion, the words we use are outer membrane, inner membrane, okay? There's a space in between the two membranes. This region here, that's called the intermembrane space, okay? And the innermost chamber, that region here, is called the mitochondrial matrix, okay? Outer membrane, inner membrane, intermembrane space, so between the two membranes, and then the mitochondrial matrix. Cool so far? All right, solid. So I'm not worried about you knowing this term here. The folds have a special name. Everything has a special name. It's the sciences. Okie doke. Moving forward, here is an image which makes my illustration entirely meaningless. But repetition is key, and I also don't write on the board enough. You know, it makes me feel lazy if I don't write on the board. I have to feel like I'm working hard. Check it out. Look at this diagram of a mitochondrion. All right? Look at this actual image of a mitochondrion. This is a thin slice through one, so you can see the folds of the inner membrane. So, outer membrane, inner membrane, intermembrane space, mitochondrial matrix inside. And within the mitochondrial matrix, check this out, there's some DNA. The mitochondria have their own genome, all right? They have their own DNA because they used to be free-living things. And if you have DNA, what's the organelle that you better have? Ribosomes. If you don't have ribosomes, you can't turn the DNA into anything. DNA is pretty useless on its own. Now, RNA can have uses. RNA can be beneficial. But DNA as a molecule isn't very useful beyond information storage. Please. They are not membrane-bound organelles. That's the key distinction. So they're the only organelles that are shared because they don't have a membrane around them. Technically, an organelle is just a complex of different macromolecule types that carries out that has an emergent function. So it's got RNA and it's got protein in it. And the RNA alone couldn't do this, and the protein alone couldn't do this. Together, they have an emergent function, a new function, and that's sort of the defining characteristic of an organelle.
So then the ribosomes would have developed on their own before the eukaryotic cells developed the nucleus?
Yes, it's possible that ribosomes evolved before cell membranes were around, you know, because RNA can make more RNA. It's all very weird, but the consensus about that stage of evolution, of molecular evolution, there are all these different molecules competing for atoms as a limited resource because some atoms are in lower supply. RNA ended up being the molecule that could best catalyze its own reproduction. And then you could see, oh, well, this starts to make these RNA structures that then could maybe assemble amino acids and make a protein, which is weird to think of. But then once you get ribosomes free floating in the environment, now the RNA rockets off, starts to make proteins in proximity. You know, this takes so much time. and isn't fully understood. But it's kind of cool to think about, you know? Yeah, great question. I love it. Cool. So yeah, you got DNA, you got ribosomes. Solid? Once again, more evidence indicating the free-living nature of these. Now, you want to get really wild with this? Check this out, all right? This outer membrane and this inner membrane are slightly different in their composition, and it is likely that this outer membrane's origin was initially the food vacuole, okay? The food vacuole ended up being the outer membrane of the ancient mitochondrion during the endosymbiosis stage. I didn't hear what you said, but I'm assuming it was 100% correct.
I said bacteria.
Yeah, it works. Yeah, absolutely. It's the outer membrane formed from phagocytosis of this free-living organism. You see, they're different in their structure, but that's beyond our points here. Fun mitochondria stuff. Okay, I want to talk to you about fun mitochondria stuff. Mitochondrial inheritance patterns. So, sperm cells are smaller than egg cells. You're aware of this. Everybody here has watched Look Who's Talking, right? Look Who's Talking Now, Look Who's Talking 2, John Travolta, Bruce Willis. Bruce Willis voices a baby. The movie starts with a bunch of sperm floating through. It's really creepy. It's how I learned what sperm cells were as a child. And they're all zooming in at an egg. And then Bruce Willis is voicing this baby. It's like, hey, can I get some milk in here? Hey. It's a weird movie. They made 3 of them, all right? In the 3rd one, the pets start talking. So, you know, learn your cinema history. It's a garbage film. It's called Look Who's Talking. Yeah. You know, it's creepy. I'm not even sure where it'd be streaming. Yeah. This is great. Yeah, totally. Watch Look Who's Talking and report back to me. Let me know how it went. You found it? Cool. It's real. Anyways, thank you. At the beginning of Look Who's Talking, you get to see 2 scale the size of sperm cells versus egg cells. Sperm cells are small, egg cells are big. Here's the thing. Egg cells being larger, they've got a bunch of mitochondria in them. Sperm cells, which are basically just designed to deliver this small package of DNA right here, okay? They have mitochondria in order to whip the flagellum that lets them speed through the fluids, okay? But those are cordoned off into this basal structure attached to the flagellum. And when the sperm cell contacts the egg cell, it basically just fuses this portion, this head portion, with the outer membrane of the egg cell and dumps the DNA in, right? So the paternal mitochondria never get introduced into the developing zygote, meaning all of your mitochondria are inherited from the egg cell. So we would say that the maternal side, right, donates all of the mitochondrial DNA. So the DNA in your nucleus is a mix of the paternal and maternal sides, the sperm DNA and the egg DNA, right? But all of your mitochondrial DNA, and therefore all of your mitochondria, were inherited from the maternal side. Okay? Question, please.
Slightly off topic, but does the same thing apply to plants?
Yes. Same thing applies to plants with chloroplasts, right? Yeah, and mitochondria. So mitochondrial and chloroplast, and it's super important for talking about disorders that happen in mitochondria and chloroplasts, right? Inheritance patterns are entirely different. We'll talk about it again when we get to our genetics section. Satisfying answer? Awesome, yes.
I was about to say, is it the same, does the same thing occur with chloroplasts when plants reproduce?
Yes. Yep. Mitochondria, chloroplasts, same sort of thing, right? As long as you have, it's not isogamy, it's heterogamy? Somebody remember from their Botany 105 that I take it recently? As long as there's small gametes and big gametes, yeah. And if you remember from Botany 105, I'm blanking. Isogamy, they're the same size. And then is it called heterogamy or anisogamy? I think it's anisogamy. Hetero? Hetero makes the most sense. Anyways, all that means, folks, is if the organisms reproduce using one small gamete and one big gamete, this happens. because there's no room in the little gamete to jam a bunch of mitochondria and chloroplasts. It's cool stuff. It's really cool stuff, I swear. Inheritance patterns, those are one of the things that can get really fun and interesting in Bio 105. Bless you. Double bless you. Even if it was a cough, bless you. All right, I'm zooming. I zoomed too far out and I've ruined everything. Don't tell anybody I did this. I'm so sorry. Okay. So check this out, right? There's this book called Parasite Eve. All right. It's a horror novel that was adapted into a bunch of video games and stuff, okay? You may have heard of the games, possibly. It's all about what if the mitochondria became self-aware and they took over the organism. You know, it's all based on this science of how mitochondria were free-living things. And what ends up happening, if this was to happen, here's what would happen. There would be an evil scientist who created a perfect offspring where the mitochondria were the intelligent controlling organisms. And then she would walk into a room and control your mitochondria and make you carry out cellular respiration at a crazy rate, and you would burst into flames and perish, you know. So that's what Parasite Eve's about. But it's all based, like a lot of fun horror and sci-fi, you know, it's all based on some true scientific principle, which is mitochondria used to be free-living things. Hopefully they're not bitter about it, you know? Because if they get upset, we're all on fire. All right, there's the mitochondria part. I love that. It's fun stuff. Chloroplast. Similar, all right, in a lot of ways. The reaction they carry out's very different, but in their structure and their inheritance patterns, a lot of the same principles apply. So check this out, right? Also a super folded up membrane. The difference with the chloroplasts is they have an outer membrane, an inner membrane, and then another membrane, all right? So instead of just having an outer and an inner, they have an outer, an inner, and then something called the thylakoid membrane, all right? So the chloroplast is the photosynthetic organ. They contain the pigment chlorophyll, a bunch of other stuff for photosynthesis. Excuse me, I'm so sorry. All right. Don't worry about plastids. That's a discussion for your botany course, okay? The different plant organelles. Just keep it simple for Bio 105. Yes? Oh yeah, the thylakoid membrane, right here. And there's the spelling of it. So you've got the outer membrane, the inner membrane. Between those two membranes, you have, once again, the intermembrane space, all right? But the difference is that there's another membrane floating in the central chamber here. So once again, like if we put another membrane, we've got more division. So if we have more divisions, it's like the house. We need more names for the different rooms. So more names, here they come. From the outside, outer membrane, intermembrane space, inner membrane, and then this big chamber here, the fluid with the fluid that is filling it, right, is called the stroma, all right? The stroma is this goo that is inside of the inner membrane. And floating in that goo, in that stroma, it's described as like a thick, syrupy substance, all right? Floating there, you've got the thylakoid membrane. The thylakoid membrane, all right, is all stacked up into these pancakes. Here's an actual image of it, okay, that are called grana, or granum in the singular. I won't test you on granum, grana, but you do need to know about the different membranes. And then inside of the thylakoid is the thylakoid space. All right? Outer membrane, intermembrane space, inner membrane, stroma, thylakoid membrane, thylakoid space. A lot of chambers, okay? And once again, the very, very folded membrane, okay? The folded membrane is where the energy processing occurs because it's full of proteins and enzymes and all sorts of things that are responsible for the energy transformation. Okay, yeah, please.
That blown up black and gray thing, that's a stroma, that's one of the thylakoid things.
Oh, this is a whole chloroplast. The arrow's meant to be pointing at the stroma where the thylakoid membranes are floating.
Oh, so that's like a real
Yeah, this is a real picture. This is a transmission electron microscopy image. So is it a picture? Not really, because the picture implies light and film, but listen, we don't need to blow minds on that one. Yeah, it's more to also see, like, look, the inner and the outer membranes are just jammed right together. The inner membrane space isn't as important in the thylakoid. I'm sorry, in the chloroplast. And once again, the outer membrane probably has its origins as the food vacuole membrane when the initial phagocytosis occurred. Okay, so the thing that the ancestral chloroplast, it probably already had the inner folded membrane. You know, yeah. It's all really cool stuff. It's all moderately cool stuff. Yeah, it's okay. Thanks. Now, I'm not asking much. I just want you to memorize all of this before I have a pop quiz at the end of class today. It's just a joke. I'm just goofing. What is this trying to show you? Look at this. You've got the inner membrane of a mitochondrion, and it's got all of these embedded protein complexes that are going to be essential for doing all of these processes to make ATP. You don't need to memorize any of this or understand any of this at all yet. I'm going to talk to you about it for like 2 whole weeks, okay? I'm just putting it on the slide to re-emphasize the membrane's folded because the essential enzymes and proteins involved in the key process of making ATP are all embedded in that membrane. So it folds up to make more surface area so that it can carry out more of that essential function. Does that make sense? Does any of this even make sense right now? All right, cool. Photosynthesis, okay? Check this out. That thylakoid membrane is going to be where all the chlorophyll is, okay? So it's going to make it way better at capturing light energy if it folds up that membrane where all the chlorophyll is so it can have more of it. All right? There's also going to be a bunch of this exact same process happening where protons get pumped and then the protons generate a proton motive force, which gets harvested by an enzyme called ATP synthase, which makes ATP. But don't worry about memorizing any of that right now because we're going to have to memorize it all in a few weeks anyways. So anyway. Peroxisomes. Boy, oh boy, do I ever hate peroxisomes, okay? They're all over the place. They drive me up a freaking wall, okay? Because the only information that you can usually find about them is they catalyze a lot of chemical reactions, okay? And the main thing where they get their name from is they get hydrogen peroxide produced after they do the reactions, like a byproduct of the reactions they catalyze. All right. So there's this enzyme in your body called catalase that breaks down hydrogen peroxide. It's the only reason I think any of this is interesting. I'm not testing you on this thing, okay? You look into peroxisomes if you want to. They're nebulous. They're too complicated because they carry out too many reactions. All right. Here's the interesting thing about peroxisomes. Your body's making hydrogen peroxide in it all the time, right? But it's really good at breaking down hydrogen peroxide without getting any harmful compounds. This is why you can use hydrogen peroxide if you cut yourself, get a wound or something, all right? It's intense, and it's like a negative byproduct. It's like toxic, right? But your body has all of these enzymes that are designed to break down hydrogen peroxide anyway and make it no longer harmful. So that is what it is. But then again, you want to think about hydrogen peroxide and how it affects your cuts and your scarring patterns and stuff like that. Just think about it, look into it, decide what you want to use. Anyways, that's that, okay? Thrilling, I know. I just can't not talk about them a little bit. In a plant cell, where would you find DNA? I should have not thrown up the answer, but DNA in a plant cell is present in the nucleus, in the mitochondria, and in the chloroplasts, okay? There's mitochondrial DNA, there's chloroplast DNA, there's nuclear DNA. Question?
So during the process of generating proteins and stuff, does DNA, like the molecule never physically leaves, like just in the case of the nucleus, it never leaves the nucleus?
That's correct, unless something went really wrong. So there's actually, and it also should never leave the mitochondria or the chloroplast. So there's actually a bunch of enzymes inside of your cytoplasm, the goo that everything else is floating in. They're nucleases that are designed just to destroy DNA, all right? But they can't get at your DNA because it's all chambered up. And this is great because then if a virus comes by and tries to inject DNA into your cell, those enzymes just encounter it and go break it down. So nuclease enzymes are a thing that you're going to encounter in our lab section because we have to be very careful not to contaminate DNA samples with nucleases, and nuclease enzymes are everywhere, all over the dang place. Yeah, solid? Awesome, I love the question, thank you. I already talked to you about this, okay? But here's all of the endosymbiont evidence. The DNA looks like a bacterial DNA, okay? They can grow and reproduce somewhat independently inside the cell. The genome, if you look at its gene density, it's more similar to prokes than eukes. There's a double membrane, all right? We got a double membrane and a double membrane. I doubled up on the double membrane. How ironic, okay? Endosymbiont theory, all right, boom, boom, boom. We're not going to talk about that. We're not going to talk about that. We don't have time. Okay? We actually, was it you who brought this up? Somebody brought this up to me at the end of the last lecture. It was you? That's right. Awesome. Thank you. Yeah, the nitrogen fixing organelle. So you've got these, this is really a fascinating article. It's super cool. You can look it up if you're very interested in it. All right? Organelles, we've talked about them. Have we talked about nitrogen fixation at all? I don't think we really have. Why would we? Why would we talk about that? Okay? Anyways, nitrogen's important. Nitrogen fertilizer allows us to grow a lot of plants. That also leads to issues with depleting the soil of other resources. Okay? This is just a cool article because it's evidence for a new organelle. Okay? Animals can't fix nitrogen. Plants can't fix nitrogen. They depend on bacteria that can fix nitrogen. Fixing nitrogen is when you can pull it out of the atmosphere and convert it into a useful form, all right? There's plenty of nitrogen in the atmosphere. N2 gas is the most abundant thing in the atmosphere, and we all need nitrogen desperately. If we could access that nitrogen without having to eat food, all right, that'd be a huge benefit for us because we'd be able to make more proteins, all right? Nitrogen is important in protein and nucleic acid construction and generation. So there's evidence for a new endosymbiote becoming an organelle. They're saying it goes beyond symbiosis, where it might start to become an obligate thing, and they live together, and as the organism reproduces, it has the capacity for nitrogen fixation. 2024 article in Science. Pretty cool. Enough. Lynn Margulis, all right, no longer with us, but the original sort of proponent of the endosymbiont theory, mocked, derided, all right, for the theory. People said, that's ridiculous, you are dumb, get out of here, I hate you. But she persevered, and now her theory is widely accepted. She did get to, thankfully, live to see her theory gain wide acceptance, you know. And I've also published for you on the PowerPoint slide a great review of all of the different endosymbiont theories that were proposed and how they actually competed, because it's never as simple as there was this one scientist and she said that this happened and it happened in this one way and nobody agreed and everybody had the same theory and her theory was the only one, you know? People had thoughts about how endosymbiosis could have occurred. If you're genuinely interested in it, this is a pretty digestible review article. You can find it, free access on Canvas. It's good, okay? It's good. And it'll tell you all about, like, how do the eukaryotes arise? Use some of your free time. I know you got plenty. It's a joke, it's just a joke that I probably made before. Anyways, enough endosymbiosis stuff, right? Are we cool with endosymbiosis? I went a little bit heavy on the PowerPoint this semester, okay? Remember, they've got their own genomes, cool, different genomes. These are like genome maps that show where different genes are. It's all fascinating stuff. Fine. Okay, moving forward, the cytoskeleton. So far we've talked about all the endomembrane system, all the membrane stuff, but we haven't really talked about the cytoskeleton at all. The cytoskeleton, one of the functions it serves is just like a skeleton, okay? It gives structure to the cell. There are three main types there are three categories of structures that are all cytoskeletal elements. It's a clunky sentence, but I said what I meant to say, okay? The cytoskeleton consists of microtubules, microfilaments, and intermediate filaments, all right? They're all made up of different types of proteins. Actin, tubulin, myosin. Perfect. Okay, good. I just wanted to double check myself before I double wrecked myself. All right. I don't know. It's Friday. Microtubules, microfilaments, intermediate filaments. They're classified, they have different structures, okay? Microtubules are like little tubes. They're hollow. Microfilaments are very thin, and they are like filaments, like a string or a cable. It's like a long thread, multiple long threads of protein that are wrapped around one another, which gives additional strength, because when you pull on a filament, the force isn't just distributed straight across, like when you're pulling on an actual like fiber, or yeah, when you're pulling on an actual like thread, just a pure string, like a pure line of beads, the force gets redirected. So microfilaments are very thin filaments, okay? Intermediate filaments are like multiple wound up fibers. So the microtubules are the biggest because they're hollow. The microfilaments are the smallest because there's just a couple of protein threads. And the intermediate filaments have multiple threads, all right? So they're the intermediate diameter, intermediate range of sizes. Here is a cell with all of the different cytoskeletal elements stained so you can see them. They're all over the place. What do? The microtubules, okay, check it out, consists of two different types of proteins, alpha and beta tubulin, all right? They get joined, they dimerize, meaning that you get an alpha and a beta joined together, all right? And then those kind of associate to make this big tube, all right? I'm not going to test you on a ton of these details because it's mainly just broad generalizations. I want you to know the cytoskeleton exists, and we're going to talk about its overall purpose, less focus on memorizing all the differences between these different types of cytoskeletal elements. Like there will not be a question where I ask you, how wide is a microtubule? Yeah, it'd be silly, I think. So what are they useful for? Okay, they are useful for maintaining the cell shape, cell motility, chromosome movements, You're gonna see a ton of overlap between these, right? A lot of these are gonna be like maintaining cell shape, all right? Yeah, so don't worry about memorizing these entire tables, okay? I just wanna show you a picture of them, give you the textbook materials. Okay, so microtubules. Flagellae and cilia, okay, are controlled by microtubules. There'll be like a special complex of microtubules at their base. The structure of cilia and flagellae are also, very different and very interesting and folks as a study on their own, okay? But you know what a flagellum is. You've seen Look Who's Talking, the movie, right? You've all seen that movie. You can see the sperm swimming, okay? Cilia are also important for moving fluid. A lot of single-celled organisms will use cilia, which work kind of like fish fins, or wings is better, where they have this thing, or they make a recovery stroke that minimizes resistance so they're not pushing themselves down, all right? Because if you just went like this, you would go down, then up, then down, then up, right? So instead, you have this like recovery stroke where you bring up the cilia, and then a power stroke where you go down, and that moves things directionally. Yes?
When
Oh yeah, totally. So at the base of a flagellum, at the base of a cilium, all right, there's like a complex of microtubules, and they sort of give a foundation to the structure. And then there will be different types of filaments running down it that give the cilium or the flagellum its actual physical mechanical structure. But at the base, there'll be this complex of microtubules that sort of give it an anchor point, and where energy can be expended to sort of make the thing move. It's like it's the anchor point. It's a pretty broad statement, vague statement. Try not to worry about it too much, because if you want to understand flagella and cilia and what controls their movement, we need a lecture, like beyond just a slide, unfortunately. Yeah, the beating just means movement. Yeah, absolutely. Awesome, cool. Okay, great. So flagella, cilia, they move differently, they're structured differently. Cilia are also useful on, say, like your trachea, okay, where they beat up like this and keep pushing mucus up so that it starts to come out of your mouth, and that captures all the gunk that you breathe in, So they're not always for movement. Sometimes they're for moving a fluid, not moving a cell. Anyway, this is more relevant to our actual course of Biology 105, because we're focused on subcellular stuff a lot more than the movements of single-celled organisms or even gametes. All right, so everybody loves this motor protein walking. So check it out. You got this special protein. It's carrying a vesicle. That vesicle has all of these little labels coming off of it that are sort of directing which motor protein it will attach to and directing it to where it's going to go, all due to modifications in the Golgi apparatus, probably. So you've got this, oh my God. See, I am not logged into my YouTube. This is just default YouTube. Great. All right. So you see the vesicle being moved and walked along the motor protein. Everybody loves it. So we're just going to pause and close this. There we go. All right. That would be really nice. I don't have the energy to do it. Or the time. Or the, what's that called? Will. Inclination. Willpower. Yeah. My give-a-damn's busted. All right? So check this out. You've got this microtubule, okay? It's serving a function of providing structure to the cell. Also, the microtubules serve as like highways throughout the cell, okay? The example given in every textbook that I've ever read is, imagine one of those New York skyscrapers being built. Those buildings, they nearly touch the sky. Ooh, ah. And you've seen the pictures of the people, and they're working on the skyscraper, and they're sitting there, and they're eating their lunch while they're just a moment away from death, And then there'll be people who go like, I'm going to go deliver some supplies. And they walk along the skyscraper carrying things, and then they deliver the supplies. So the microtubules provide structure, and also you can move things along the microtubules. Make sense? The analogy there, cool? All right, awesome. So motor proteins can attach to these things and walk along them. This is work, moving stuff, so it requires energy. The energy is in the form of ATP, which is the basic energy currency of the cell in general. And that's not what I'm trying to do. I'm trying to skip to the next thing. Thank you. Oh my, everything's so rough. It's so difficult to be me. You cannot even imagine. All right, check this out. Here's an actual image, okay, of things being moved along these microtubules. The microtubules have been stained. You can see the vesicles moving around. It's a pretty active process, right? It's pretty cool. And based on how the vesicles have all, or the vacuoles, have all been modified by the Golgi apparatus. You can determine which motor proteins they'll attach to, which determines where they go. So hopefully you start to see now, oh, how could we actually direct certain protein products from the endoplasmic reticulum from ribosomes to go to certain locations? It's all about like carbohydrate labels, which motor proteins can attach, etc., etc. We can just come back to these last few slides on Friday because we're going to talk about the other two types of cytoskeletal elements, and then we'll talk about cytoplasmic streaming, and then what's outside of the cell, and then we'll finally be able to get into the cell membrane lecture. All right, that concludes it today. Have a good rest of your Friday. Have a good weekend. Let me know if you have any questions, and I will see you all again on Monday. Thank you. You too. Have a good one.